I'm Wang Xin from Professor Lin's lab. My research interest was mainly focused on the biogeography of syngnathid fishes. Now I will play the recording video. Just wait a minute.
Hello everyone, I'm Wang Xin. I come from South China Sea Institute of Oceanology, Chinese Academy of Sciences. I'm very glad to be part of this meeting and share my latest research progress. Well, the topic.
Wait a minute.
Hello everyone, I'm Wang Xin. I come from South China Sea Institute of Oceanology, Chinese Academy of Sciences. I'm very glad to be part of this meeting and share my latest research progress. Well, the topic of my presentation today is phylogeographical patterns of syngnathid fishes in Northwest Pacific. Here is my main contents of my speech. The syngnathid fishes are amazing animals. They acted as iconic species in many research areas, including the conservation of marine biodiversity and the adaptive evolution of many complex traits, animal behavior, and even bionic researches. The syngnathid fishes is one of the most successful groups of the global radiation and dispersal. The coral triangle maintained the highest species diversity, which is regarded as the marine biodiversity hotspot. Well, the Chinese coast is an important channel for their northward dispersal. So the first question is, how many syngnathid fishes are distributed along the Chinese coast? In order to answer this question, we made a lot of field investigations in recent years through scuba diving and fishing port survey. And we also got some data from the customs and agricultural sectors. Now we have got some preliminary results. Combined with the literature records, a total number of 56 species that belong to 26 genus were recorded, distributed along the Chinese coast, including a new seahorse species reported by us. Additionally, we also found several new recorded species in local areas, such as the straight stick pebfish. We found that the straight stick pebfish are distributed along the north coast of the South China Sea, and it has obvious morphological differences with its relative species, especially in the snout lines. And we collected the straight stick pebfish samples from 3 sites along the north coast of South China Sea, including Kending, Zhuhai, and Beihai. We firstly extracted the DNA and amplified the mitochondrial CYTB genes. And the population structure analysis revealed that the Beihai population was a relatively independent lineage, and the FST value between Beihai and other 2 populations was relatively high. Well, this might be explained by the historical changes of Lezhou Peninsula, because when the sea level falls in the history, Lezhou Peninsula was a land bridge which will cut off the population exchanges in some extent. And these kinds of barriers were commonly exist in the Chinese coast, like Dongshan Land Bridge in the Taiwan Strait and the Changjiang diluted water. So the question here is, what is the phylogeographic patterns of the parrotfish populations like in the Chinese coast? In order to answer this question, we selected a widely distributed seaweed parrotfish as our research object. Firstly, our simple sites cover the coastal waters of Yellow Sea, East China Sea, and South China Sea. The morphological and genetical differences were investigated in this study. We conducted the morphological analysis with a geometry method. Firstly, we selected 26 cranial landmarkers and 163 specimens were used in this study. The relative warp analysis shows that the three populations have significant differences in terms of relative warp 3. The relative warp 3 represents the eye size. The specimen plotted on the positive axis of relative warp 3 tend to have larger eyes, whereas these on the negative axis have small eyes. Well, this kind of differences between different populations may related with the beta type and turbidity of water, maybe. As for the population genetic structure analysis, we sequenced 69 specimens from the Yellow Sea, East China Sea, and South China Sea. And the restriction site associated DNA sequencing method were used. And finally, we got 90,000 SNPs for the phylogenetic analysis. The genetic results showed 3 distinct clusters that represent the 3 geographical linkages. And also you can see that the South China Sea populations formed a separate cluster. And what's more, the space distribution model suggested a clear geographical separation between the South and the North populations during the last glacial maximum. Well, this part of work was analyzed by Dr. Zhang Zhixin from Hokkaido University of Japan. And the ecological niche characterization with hypervolume indicated that the 3 clades diverse in their niche across their distribution ranges. This part of work was also analyzed by Dr. Zhang Zhixin. The results of ecological niche divergence and the history distribution of seaweed parrotfish were identified with the genetic divergence of seaweed parrotfish population. And the further genetic analysis between the different seaweed pupfish populations found that many genes were identified with strong selective sweep signal, which are important to determine the genetic basis of local adaptation affecting fitness traits, including the genes involved in growth, cold adaptation, and eye development. Finally, to sum up my presentation, we got two main conclusions. The first is the diverse habitats along the Chinese coast maintain a relatively high syngnathid species diversity. And the second is the phylogeographic pattern formation of the seaweed pupfish could be explained by a combination of historical geography, ecological niche divergence, and the risk of local adaptation. And finally, I want to thank for the foundations from NSFC and the GML, and thanks for the help of our research group. Thank you for your listening, and the questions are welcome.
All right, thank you for your listening. So someone have a question? All right. All right, this is the last speech. So thanks for all the fantastic speeches tonight in the section of systematic and evolution. I believe everyone have a good time in this section. Okay, thanks for your time and bye bye.